So at this time, we're fortunate to have from Sanofi, Dr. Luis Felipe Orozco Cabal. And Luis, I really appreciate you being here to offer a public comment. You're on.
Thank you, Professor, and thank you for the opportunity of commenting today. First and most importantly, we would really like to express our deep gratitude to people living with MS who participated in all the clinical studies of the investigational agent tolebrutinib and all our clinical trial programs. We also want to thank their support networks, including caregivers, friends, and families. Their willingness to participate in this clinical and critical research allows for breakthroughs and innovative science to be driven forward. So let's talk about the current situation. So as it's been stated before, current MS treatments focus on controlling relapses and acute lesions, but people with MS continue to experience disease progression due to the smoldering neuroinflammation. While patients on current MS treatments have the relapses under control, they're continuing to have disability progression. And this leaves a significant unmet need, particularly for people with non-relapsing SPMS, for whom no approved therapies are currently available. Some of the people with non-relapsing SPMS interviewed by Sanofi as part of our development process shared that this decline is relentless and persistent. And this has been mentioned all throughout this meeting. Each year, people living with this form of MS, they lose more and more function and become further reliant on caregivers. And not only Sanofi sees this, I think the FDA also recognized this high unmet need and granted breakthrough therapy designation to tolebrutinib for the treatment of adults with non-relapsing SPMS. The drugs qualifying for this designation must show preliminary clinical evidence that the drug may demonstrate substantial improvement on clinical significant endpoints over available therapies. If approved, tolebrutinib will be the only MS treatment that crosses the blood-brain barrier, exceeding CSF levels necessary to modulate immune activation inside the CNS, directly addressing the drivers of neuroinflammation and neurodegenerative processes within the CNS, and that subsequently lead to disability accumulation in these people. Phase 3 results from Hercules clinical trial in people with non-relapsing SPMS show that participants treated with tolebrutinib experience a 6 months confirmed disability progression risk reduction of 31% compared to placebo, as Dr. Lean described before. Also, liver enzyme elevations above 3 times the upper level of normal were observed in 4.1% of the participants receiving tolebrutinib, compared with 1.6% in the placebo group. A small 0.5% proportion of participants in the tolebrutinib group experienced peak ALT increases 20x upper level of normal, all occurring within the first 90 days of treatment. All but one case of liver enzyme elevations resolved without further medical intervention. And prior to the implementation of the revised study protocol with more stringent monitoring, one participant treated with tolebrutinib experienced hepatotoxicity, leading to liver transplant and died due to postoperative complications. Once we identified a signal for hepatotoxicity in mid-2022, the stringency of monitoring was increased, such that weekly monitoring was implemented between weeks 2 and 12, with monthly monitoring from month 3 until month 12, and quarterly monitoring through the end of the study. Now, given the lack of any other approved treatment option for people with non-relapsing SPMS and the substantial unmet need that ICER acknowledges in their report, Sanofi feels the pricing recommendations laid in this report do not reflect the full value and innovation that this treatment represents for the full, should it be approved. Despite this being the first in disease treatment, They are recommending prices well below what we recommend for new therapies in relapsing forms of MS, while the unmet needs still exist with relapsing forms of MS. While the unmet need still exists within relapsing forms of the disease, the way ICER is assessing relatively values falls short. It penalizes those innovating in the new space versus those innovating where there are alternative options. A robust value-based pricing system would reward both first-in-class and best-in-class innovation. It would not penalize first-in-class innovation relative to best-in-class innovation. Thank you for this opportunity and thank you for your attention.
Thanks, Luis, for your presentation. We actually, we have some time if our panelists would like to ask Luis some questions or any further elucidations. Lisa?
Yeah, thanks, Dr. Cabal, for your, for the work that Sanofi has done in trying to find a treatment for this unmet need, as we've talked about. And also for doing what seems to have been a pretty extensive study, trying to assess the efficacy of the drug. Given that and the discussion that you may have been listening to, and in the fact that you guys did do the very extensive quality of life 54 question scale, I was wondering when the results of that and the cognitive function, as well as the results based on the pre-planned stratification, are going to be presented, if ever. Just noting that if those results aren't presented, I think we're all going to be left feeling that they must be negative and potentially even showing in the other direction, showing not only there was no benefit, but maybe things were worse with the drug. And again, I think the data is there, so we hope there's going to be a chance to see it at some point on some of those measures. So just thoughts about when that might be available.
Yes, thank you, Professor, and thank you for the question. Very important one. As you say, our clinical development program with tolebrutinib is very robust. It's actually one of the largest we've ever done. And as you can imagine, the amount of data that has been collected is quite large. So analysis of the data are on the works and will be presented as they become available on the next congresses. Now, some of the data, and you know how it works with the congresses, so you submit all the abstracts, there's a period of time in which the data is embargoed till we present the findings, but all of those findings will be presented during the upcoming congresses. Also importantly, as you say, we did use MSQOL 54, and the data will be presented shortly in the next few congresses.
Thank you. Sorry. And I'm not sure, do you mean like national meetings or things? Like, what's the time frame? A year, 6 months, 2 months?
As they become available, I mean, the time for the analysis takes time, right?
Right.
But it will be presented. I mean, it's also in our interest to present this data to the community, right? And to the scientific community. So,
I'm sure as a company, you have very specific deadlines for when it has to be analyzed, but it sounds like not able to share that right now.
Exactly. It's on the work. The analyses are there, and it will come. We have the next big meeting is ECTRIMS. So ECTRIMS, some good amount of data will be presented there. Yeah.
Thank you.
Bob?
First, a hearty warm fuzzy to all my panel colleagues for being so patient, focused, and oriented with your questions. I really appreciate how far we've come over the years with this. Luis, I would give you a hearty pat on the back for even being willing to come here and speak up, since so rarely the drug companies actually do show up for these things. I am aware of your position and limitations, so I'm going to try to put you on the spot in a little bit obtuse way. BTK has been around for a very long time. It's not a new thing. BTK inhibitors appear to have been a relatively recent focus. And here comes the put on the spot thing. MS is a lot smaller than oncology and future profit. I'm very curious why we're going at MS first. If you could speak to that a little bit, and I realize you're probably not going to be able to say a damn word about it, but what's in the future for this drug beyond MS?
Yes, Robert, and thank you for your question. Again, as a company, it's very important to be participating in these forums, and it's a great opportunity for us to be part of these discussions. With regard to the BTK, you're absolutely right. BTKIs have been used in oncology before. They're being used quite and researched in the oncology field. Within the neurology franchise, we have been very, very interested, having been in the field of MS, of understanding better the underlying mechanisms or the biology of the progressive forms of MS. And that's our keen interest in understanding how can we find targets and molecules that can cross the blood-brain barrier and actually act on that chronic neuroinflammation that seems to be the main mechanism behind progression, right? So tolebrutinib was particularly designed to cross the blood-brain barrier and be able to have good target engagement, basically focused at the microglia and innate or the living B cells living within the CNS that could actually change the biology of disease progression. I also have to say that BTKIs and particularly tolebrutinib is teaching us about a very different process within the brain. It's teaching us, many, many things about progression. So it brings a new story, and as it has been mentioned before, it's a changing paradigm in the way we think about MS, right? For the last 20 to 30 years, we have been focused on relapse control. If you had worked like 30 years before in MS, we were previously focused a lot on disability progression. Then we moved into controlling relapses. And now we see that we have, let's say, good clinical results with relapse control, then we're also able to uncover what's going on with disability progression, right? So I think this is a big, big shift in the field. Our commitment as a company with MS is really to continue trying to defy disability progression and trying to find ways, because we know it's an area of unmet need. And that's for our commitment with MS, with neurology, and with the community.
Thanks, Luis, and thanks for that question, Bob. I'm going to channel Rita Redberg on what she would be asking. I'm thinking sometimes I can sense her concern, particularly related to randomized clinical trials and lack of representation in key subgroups. So and you please correct me, and first of all, I've been involved in clinical trials. I know how difficult it is related to recruitment in key groups. So please correct me if I have this wrong. The patients were relatively young, average age of 49. Underrepresented group was African American. And I think a lot of the unmet need may be in the older patient related to this particular discussion with this particular drug. Any comments on that, Louise? Efforts that we're going to be looking at and getting in those key groups so we can assess efficacy in those patients?
Yes, I think that's a really good question. Traditionally, the trials in MS have been done usually in population that is less than 55 or 60 years. I think we have a little bit there of, I think we need to think as a field, as our populations are aging, and actually you see patients now in their 75s and 80 actually with MS, and that's only the result of our success in treating this disease, right? So traditionally, the phase 3 studies involve people less than 60 years or 55 years. I think now we have a better idea of the relationship between aging and MS, and I think it's really an area where we need to do more research. So, it's part of the evidence that we still need to collect.
Yeah, thanks, Luis. Aneta.
Yes, I want to echo my colleagues' comments here on the panel that thank you so much for moving into this space and your company's commitment, not only with tolobrutinib, but also with the frexalimab trials as well. With that, though, I have actually been told by some of my colleagues who have attended your ad boards that have been presented the data stratified by gadolinium enhancing lesions to the active and inactive subgroups. So I'm just kind of wondering, when the rest of the neurology community would be able to see that.
Yes, Dr. Langer, that's a really good question. Again, yes, we started doing the subgroup analysis. Again, this data are embargoed and we expect to present it at ECTRIMS. That said, so I hope you stay engaged for the release of the data at ECTRIMS. That said, I think there's something really interesting about tolobrutinib and the other BTKIs that have been researched thus far is many of the ground truths that we had, and I think this is triggering also a discussion around how we design clinical trials in MS, are challenging what our outcomes actually mean in this population. This is a population that has not been studied thoroughly. We're starting to understand the biology of SPMS. Many of the, let's say, the outcomes that we designed and measure for RMS, I'm not completely sure we understand. They mean exactly the same in an SPMS setting, right? So stay tuned for the results, but I think that the most relevant thing around those results is the discussion that we have as a scientific community around what are the best outcome measures and what are the imaging biomarkers that we can use in this population. As you know, in addition to GAT1s, which are typical from the, let's say, relapsing biology, there's been a lot of discussion around PRLs, right? So we're also seeing, and we have reported on very interesting findings on PRLs in patients from the Hercules trial. And again, it's just it's super interesting to see how that biology manifests in this imaging biomarkers that we have. So I hope you stay tuned for that. I think it's going to be very thought-provoking. And I think, we're advancing the field. It's a conjoint responsibility with the scientific community and us as manufacturers.
Luis, again, thank you so much for being here today. Thank you so much for sharing your thoughts and taking on some tough inside slider questions from our group. You handled them beautifully, so terrific. At this time, yeah, really. At this time, I'd like to take our lunch break, and for the people on the West Coast, we'll be returning at 11:50 a.m. Those on the East Coast, you'll be returning at 2:50 p.m. Eastern Time. Thank you very much, everyone. Talk to you later. Have a nice lunch.